So this is lecture 7 in a series of 8. We've been examining over the past 6 lectures issues that are important in the design, conduct, and analysis of clinical trials. We start out by talking about why we do clinical trials in the first place, the challenges of questions and the measurements we use to answer those questions, some basic trial designs, how you decide how large a trial should be, and last time we examined in some detail how trials are monitored for safety and benefit. You may recall there's a need to have a monitoring plan in place for all trials, especially if they're phase 3 trials. You may need to have a special committee, and there's some statistical issues that have to be kept in mind in the process of monitoring. The more you look, the greater the chance of finding something by chance alone. Today we're going to examine some issues in the analysis of a trial that have to be kept in mind. A couple of key references about analysis issues I provided here that give you some of the examples that I will use in this lecture. One of the key questions in a clinical trial is what data should be analyzed. We're not going to talk about the statistical methods. That's a time for another course. But we'll talk in some detail about the philosophy of analysis. There's the idea of the intention to treat principle, which really means that you analyze what is randomized, as I've indicated, all patients, all events that take place. There's a lot of trials which claim to be intent to treat, but in fact, if you look carefully, they're not exactly intent to treat, and they differ in ways that might be very important. When we talk about what data to be analyzed, we're not talking about the patients who were excluded prior to randomization. Of course, that process affects generalizability, as we discussed in our earlier lectures. But what I'm concerned about is those patients who are withdrawn from the analysis. They were randomized, but for some reason, valid or not, are not included. As soon as you start excluding patients from your study that you've randomized, you have the opportunity to induce serious bias. We've worked hard over the past lectures to point out ways to remove bias from our study, from how patients are allocated, how they're evaluated, and so forth. But it's quite possible to have a perfectly designed and perfectly conducted trial and to really introduce major bias at the very end, something we want to avoid. There's a general principle of intention to treat, which is defined in the International Council on Harmonization document in section called E9. They define intent to treat as has the consequences that subjects allocated to a treatment group should be followed up, assessed, and analyzed as members of the study, irrespective of their compliance with the planned course of treatment. Fairly straightforward, all patients, all events. However, it's common practice, was less so today perhaps, but still nevertheless it occurs, to withdraw patients from analysis. In the 1980s, there was an article in the New England Journal that examined this practice. Over a 3-year period, there was 109 trials that were included in this review, and 37 of those trials admitted to not analyzing all the data of patients that have been randomized into their studies. The reasons given sound quite reasonable. For example, the patient was ineligible in retrospect. After you re-examine the data, you decide that patient should never have been entered into the trial. The patient didn't comply to therapy as hoped for. They had some other event, or the data was missing, so the patient was excluded. Let's examine the ineligible patient. You didn't intentionally randomize an ineligible patient, but after the trial was over, you discovered by reviewing the data that some patients did not in fact meet the eligibility criteria. And your concern is that those ineligible patients may in fact dilute the treatment effect and hurt your chances of having a positive trial. So the idea is, let's get rid of those ineligible patients and withdraw them from the analysis. This process, in fact, can cause bias. Let me illustrate that with an example. This is a classic trial. The Antran reinfarction trial was published in the New England Journal in 1980, and it sort of established, in some ways, the intent to treat principle as being the principle that needed to be followed. This was a trial, randomized, double-blind, placebo-controlled, comparing Antran versus placebo, Antran being a product that reduces the ability of the blood to clot. 1,629 patients were randomized approximately equally in the two groups. After the fact of randomization, 71 patients were declared to be ineligible, 38 versus 33. Those reasons broke down into 3 groups. The heart attack, the MI, was either too fresh or too old for the therapy to ideally work. The heart attack was not severe enough, that is, the enzymes were not elevated sufficiently. Or there was some other competing risk that made the patient not ideal. 71 patients, let's toss them out, was the idea, and get rid of whatever impact they might have in the trial. They come out of the 1,629, and if you reduce or eliminate them, you're left with 1,558 patients left. There were some additional rules that were applied in this trial. There was the so-called 7-day rule, that if you were not on therapy in the trial for less than 7 days, then you didn't have a chance for the therapy to work, the drug to work, and you were not included in the analysis. So in this trial, we would throw out another 30 patients. So we go from 1,558 to 1,528. Then there was a case of analyzable events, or analyzable deaths. If you had an event 7 days off the drug, those events did not count. So you'll see in this discussion a series of decisions which are made to eliminate patients and events. What is shown in this table are the mortality results of the Antran reinfarction trial for all patients randomized, which had 74 patients dying on Antran and 89 on placebo. Clearly a trend in favor of Antran, but the p-value is 0.20, clearly not close to evidence for clinical benefit. If you were to examine the patients that were declared eligible, the 1,528, then there's 64 patients with a death on Antran versus 85 on placebo, and the p-value is a close call, or almost significant, at the 0.07 level. There was an earlier paper, actually, in 1978, where that p-value was actually below 0.05. Among the ineligible patients, which is the point of this graph, there were 10 patients on Antran that died and 4 on placebo. Because out of these 71 patients excluded because they were ineligible, there was 14 deaths, 10 versus 4. Look at the mortality rate on the placebo patients, whether you were eligible, i.e., or ineligible, the mortality rate's about the same, 10.9 versus 12.1. Clearly not significant. But if you look at the patients on Antran, there's an 8.3% mortality among eligible, a 26.3% among ineligible, a p-value of 0.001. That is quite a significant difference in the mortality rate of those who were excluded on Antran compared to those who were excluded on placebo. Details of this can be found in a publication by Temple and Pledger that was published in the New England Journal in 1980 and led to the establishment of the intention to treat principle as the preferred analysis. Let's examine this a little more carefully. We're going to look at data that was available compared to what was published in the New England Journal paper. We know that among the 1,629 patients, there was 163 deaths. P-value of 0.2 as indicated earlier. But what's featured in the paper, if you add up carefully, is 149 deaths by combining the results in two tables. Among the 1,558 patients, there were 64 deaths versus 85, or 149 deaths total. One might assume that that's the number of deaths you would see in the final analysis, because you've gotten rid of the 10 versus 4. However, that's not what is presented as the primary analysis. Among the 1,528 patients, after you get rid of the 71 ineligibles, there's the so-called analyzable deaths, which is shown in table 4. We have 44 deaths versus 62 for a total of 106. That's where the featured p-value of 0.076 comes from. So you notice we've done two things. We've eliminated patients, we've eliminated events. Now, it did not reverse trends. The trends are still favorable, but it has a dramatic effect on the p-value and the significance level. So you can see that there's a bias that's introduced by eliminating subjects based on eligibility. So, based on the Antran trial and others, we have to have some policy about what we do about ineligible patients. In some trials, you can delay randomization until you've been able to confirm eligibility, and then there's no reason for withdrawals. This was the case in a trial called the Aspen Myocardial Infarction Study, or AMOS. Many trials must accept ineligible patients because there's no time for verification, but if you do so, you must allow no withdrawals. That was the case of the beta blocker heart attack trial and another trial called MILUS, which was a trial assessing the size of the infarct in treated patients versus control patients. In any case, we should allow withdrawals only if the procedure is defined in advance, the decision is made early before the events are accumulated and assessed, the decision is independent and blinded, it's using only the baseline variables, the profile of the patients as randomized, and above all, you present the data both ways. Then we can have a discussion about whether the ineligible patients should be included or excluded, and we can have a serious scientific discussion. If the data is not presented that way, maybe you're not even aware of the additional events, and you can't have a debate at all. The second reason that patients are sometimes excluded is they did not comply. This was again, widely discussed issue around 1980. The argument that there's really two kinds of trials. The so-called management trial, where you apply the intent to treat principle. That is, you intend to treat people with a certain management principle. Some will comply, some will not. But no matter what, you're going to analyze all patients, all events. The second kind of trial is called explanatory. What you really want to estimate, ideally, is the optimum effect if patients perfectly complied to the therapy. So let's only analyze those who comply as desired. But as I've indicated here, withdrawals for noncompliance can lead to serious bias. Let me illustrate that with a very simple example. This is the cancer trial, just to shift away from cardiology for a moment. We have plotted here 3 assessments of compliance according to method 1. We compute the dose actually received of the adjuvant therapy divided by the total dose that the protocol prescribed. As it's shown here, the disease-free survival over the 5 years post-surgery, the good compliers are defined to be those with 85% or better of the dose prescribed, those which are moderate compliers, 65 to 84% in the open triangles, and those are the poor compliers, the open circles, which are less than 65. Well, as expected, those by this definition, those who comply get the best disease-free survival. Those who are the moderate compliers get the moderate intermediate results, and the poor compliers get the poorest results. But now suppose I tinker with the definition of compliance just slightly. In this slide, the method 2, the dose received is the same as in the previous definition in method 1. However, I've changed the denominator from dose as fully prescribed in the protocol. Here I define the full dose while they were in the study, while they were active. A subtle change, and perhaps a reasonable alternative definition. But notice what it does to the ordering of the outcome. Again, 85 above is good compliance, less than 65 is poor, intermediate in between. In this case, the top best disease-free survival is in the poorest compliers, opposite to the other definition. The best compliers are in the middle, and mediocre compliers are at the bottom. So a subtle change in the definition of compliance can reorder the results. That's quite worrisome. Another interesting fact, perhaps the most important fact, is that this is the placebo arm of this study. By tinkering with the compliance to placebo, I can order and reorder results any direction you would like. This is an opportunity for serious bias. There are many such examples of analyzing patients by compliance, which have indicated some results which are hard to explain. Let me go back to the coronary drug project one more time. 5-year mortality, clofibrate versus placebo. Recall the mortality curves are identical. The top line indicates 20% versus 20.9% in the 2 arms. But now suppose I examine the data by compliance, where I split the data by less than 80% and greater than 80%. Here in this case, we're counting pills. So if you examine the data on clofibrate, the poor compliers, less than 80%, have a 24.6% mortality. The good compliers, those who took 80% or better, have a 15.0% mortality, indicating that if you take the medication, you do well. However, notice results in the placebo arm. Here too, if you take the placebo, you do pretty well. In fact, you do the same as if you take the drug, clofibrate. So compliance is a confounder, and you can't unravel it. If you do try, you'll get results which make no sense. Good compliers and placebo doing better than poor compliers and placebo suggest the opportunity for confounding and bias. A second important note is that in this study, there were many known risk factors for heart disease. In fact, the author, to push the point, used 40 of them and could not explain away the difference between placebo compliers and non-compliers. In other words, we can't adjust for this confounding by the methods and the covariates we have available to us. This leaves us with the fact that we must account for all patients, all events, regardless of compliance, i.e. the intent to treat principle. Another example to illustrate how complex this is a different study, estrogen versus placebo in a population of heart attack patients. And notice here, again, splitting by compliance of 80% better or worse. In the estrogen-treated patients, the mortality outcome is the same, 6.1 versus 6.3. In the placebo patients, for the poor compliers, they had the worst results, 9.9, versus the good compliers, 4.8. So again, it's difficult to make this even more complicated to predict in which direction the analysis may go, trying to adjust for compliance. So to summarize compliance, there's no consistent pattern. The point to emphasize is that compliance is itself an outcome, and it's not always independent of treatment or how the patient is doing. For sure, withdrawing patients for their compliance can lead to bias. Non-compliers can, in fact, dilute treatment, but it's very hard to account for that, and the best we can do is try very hard to not randomize non-compliers in the first place, and then in our design and our sample size calculations to adjust to compensate for the non-compliance, as we discussed briefly in our discussion of sample size. Competing events. Here, we're talking about a trial, for example, which is following heart disease and the patient dies of cancer, or vice versa. You're doing a cancer trial, and the patient dies of a heart attack. Trying to treat a heart attack in the cancer trial, you weren't trying to treat the cancer in the heart disease trial. The methods we have available, statistically, always assume that the competing event was independent of the event or the disease you're studying. We sometimes talk about censoring in survival analysis, which clearly assumes that the censoring process, the lack of information after a competing event, is independent. But that's an assumption. It's very hard in some cases to verify. So we need to be careful when we enter patients into studies that we don't have somebody in a heart disease trial who has lung cancer or liver cancer, or a patient in a cancer trial who has just had a serious myocardial infarction. There is the problem of definitions. Let me go back to the Antron reinfarction trial. As you may recall, the results for mortality were 0.07 on the edge. This trial changed the definition of its primary outcome from the 1978 paper, which I didn't discuss, to the 1980 paper, from mortality to sudden death. Sudden death is an event which must be classified by someone, a physician or a group of physicians, and it's a matter of judgment. In the Antron trial, they declared there to be 30 sudden deaths on Antron and 48 on placebo with a significant p-value. Because of all the controversies that surrounded Antron reinfarction trial and its classification of who was in and who was out of the analysis, the FDA asked for another committee to reclassify the sudden deaths, and they came up with 28 on Antron and 39 on placebo, p-value of 0.17. Depending on whether the committee was blinded or perhaps not blinded, one has the opportunity to introduce bias in how you make those assessments. So whenever one has a cause-specific event, such as a death, one has to be very careful that the bias does not introduced into the classification process. Here I've just summarized the classification of events on all patients, all deaths, from the New England Journal paper and the other committee, versus those that they featured, the eligible patients. In either case, you can see that there's a shifting in the significance level from the article that was published versus the results from the alternative committee. Missing data, a real problem for any trial. You want to reduce your missingness. You'd like it to be zero. Sometimes it's not possible. The problem is that the missingness may be associated with treatment, or at least patient progress. For our purposes, data are not usually missing at random. There's a reason it's missing. If the number of patients missing is substantial, again, we have the opportunity to be missing an important signal. We want to implement the trial with minimum possible missingness and try to explore missingness, the impact of missingness, by various statistical techniques. But generally, it's very difficult to convince yourself that missingness has no impact. The message is design the trial with outcomes that are likely to be assessed, ascertained in all patients. During the conduct, work very hard to not have missing data. And then, if you have missing data, try to assess the results in various ways. This is just one such way, the best case, worst case. In this study, there was 170 events on treatment, 220 on control. There were 30 cases that were lost to follow-up, don't know what happened, on treatment, and 10 on control. Well, the best case analysis would be, for treatment that is, that there was no more events for treatment, treated patients, and there was all the missing data, and the control had an event. So you have 170 versus 230. The worst case would be that all patients missing on treatment had an event, and none on control. Well, you can see that these are very conservative, and not many analyses can withstand this kind of best case, worst case analysis without having differences in the analysis results. So it's not a very good way to rescue yourself. Better to not have any or to minimize. Poor clinic performance. We generally randomize trials with it's stratified by the clinic. Then if a clinic isn't able to perform, it doesn't impact the rest of the study. But withdrawing a clinic from the study must be done so very carefully. It must be done independently of how the outcome is doing in that particular clinic. Let me share with you an example. I mentioned earlier the aspirin myocardial infarction study, or AMOS. This was a trial of a very high dose of aspirin versus placebo. Contrary to expectations, overall it did not show a mortality benefit, in fact, slightly in the wrong direction. But if you allowed me to pick the 7 selected centers and look at their data, I can clearly find a significant result. In fact, p less than 0.01 in 7 of those centers. So picking and choosing centers allows you to pick and choose your result. Again, an open door for bias, something we want to avoid. This issue of centeredness, here's a trial, the beta blocker heart attack trial, which we've discussed. Remember, that was a very positive trial for propranolol versus placebo. P-value less than 0.01, very impressive statistic. However, if I pick out just the right centers, 6 of them in fact, I can show that the mortality is higher on propranolol than placebo. Significantly so, P less than 0.05. So selecting centers is something that we should not do if we get to look at the results. Finally, let me talk about an example of fishing around or dichotomizing outcomes in the data. We often, because of convenience and simplicity, take some continuous type outcome and break it down into what I will call success and failure. My point here will be that if you change the definition based on the data, let the data derive the definition, you again can introduce bias. If we're going to have definitions of success and failure, they must be stated in advance, and they must be based on external data from somebody else's trial or somebody else's study. Let me share with you a simple example I created. It's artificial, but it illustrates the point quite nicely. Here's a study, 2 treatments, A and B. I have 25 subjects on A and 25 subjects on B. I measured the patients before the treatment and after the treatment, and I took the difference in the response. Imagine in this example it was heart rate, something simple. If you look at the 4 columns of results, pre, post, in treatment A or treatment B, or look at the delta, the difference, you'll see no suggestion or evidence of a treatment effect. It's different between A and B. But you're disappointed in these results, and you say, I've worked so hard, there must be something in here I can get out of this data. Suppose you talk yourself into a definition of success and failure as a change of 7 beats. And you say, okay, let's look at the data and see what happens. If you apply that particular criteria to this data, you'll get 23 failures on A and 25 failures on B, or 2 successes on A. P-value is still not significant, 0.49. You're disappointed, but you're not willing to give up. Say, well, how about we try a different definition? Let's look at a change of 5. In this case, we have 29 failures on A and 25 on B, or 6 successes on A. P-value 0.02. This looks very interesting. In fact, my abstract might actually get accepted with this definition of success. Being enthusiastic, we say, well, how about we try an even better definition of success and failure, a change of 3. Well, here, too bad, results disappear. We again have a non-significant result. The point of this somewhat artificial and perhaps silly example is to indicate that by playing with the definition of success and failure, given that I see the results, I again can create a, quote, significant result, when in fact there's absolutely nothing going on in this data to suggest any difference in treatment A and treatment B. This illustrates a dangerous practice of making definitions after the results are known. One final comment. We have statistical methods, quite sophisticated methods, which allow us to make adjustments of covariates for outcomes. We do that, of course, to adjust for any imbalances that might happen in the randomization, and perhaps to gain some slight advantage in the efficiency or power. But the problem is, if we adjust by outcomes, which are what we call time-dependent covariates, we don't recommend that in clinical trials, despite the fact that these powerful statistical methods exist. In epidemiology, we do that because we have no alternatives. If we're monitoring blood pressure changes, or lipid changes, or exercise tolerance changes, and trying to predict risk or the impact, that's all we have to go on. So we model it in this fashion. But in the clinical trial setting, it can lead to some serious bias. Let me show, again, a very simple example, and once again comes from the Coronary Drug Project. Remember, this is a study in men who've had a heart attack, and we wanted to change their cholesterol levels using the drug clofibrate in this case. So we split the data into high and low, those with less than 250 milligram percent cholesterol, and those which are higher than 250 milligrams. We further put the data into those for whom the cholesterol improved as it went down versus those where it rose. So you look at these results, and you see in the clofibrilate-treated arm, if you were low cholesterol and it went down compared to those going up, you had a benefit, lower mortality, 16.0 versus 25.5. If you had a high cholesterol, in fact, and it fell, there was 18.1 versus 15.5 for those for whom it rose. This is an odd result. And placebo, whether you went up or down, didn't make any difference, essentially for the low cholesterol or the high cholesterol. But the fact that the best results for those patients in this study were those who had a high cholesterol and for whom it got higher is nonsense, contrary to everything we know about this. The lesson we learned from this little example is that adjusting for a covariate change is going to provide an opportunity for bias to become into our analysis. So despite statistical methods which allow us to do this, it's just not a good principle to follow in a clinical trial because compliance to therapy or response to the therapy on some intermediate marker can introduce bias. Subgroup analysis. We've talked earlier about repeatedly analyzing a particular variable, but the same concept is true if we look at lots of variables, or lots of outcomes, or lots of subgroups. The more we look at, the greater the chance, the greater the probability of finding something by chance alone. Look at one, the statistical methods would suggest a false positive rate of 0.05. If we look at many variables, it can go up to 0.19 or higher. While this is quantified from the frequentist point of view, the same issue exists whether you take a Bayesian point of view. It's just quantified in a slightly different way. So we talked about focusing on a particular subgroup can be risky because it could look important just due to chance. Results might not be consistent. And the estimates are not precise due to small sample size. So if we isolate down on the particular subgroup that interests us or looks different, we really could be looking at chance and something that's not very precise to begin with. Let me go back to the MERIT trial. You saw these results earlier, placebo versus metoprolol, an impressive 34% reduction. If you look at those results, as I said earlier, across the classic set of risk factors for heart failure patients, you see a remarkable consistency. The subgroups are generally large enough, although there's a few that are small, which have a very broad confidence interval. But there's general consistency. However, suppose we examine these results on a different classification of subgroups. Country. Regulatory agencies are always interested in how the drug responds in their country. The danger is, though, that there's variability across sites, across countries. What's plotted here is the relative risk for total mortality and hospitalization, or total mortality and cardiac heart failure hospitalization. You'll notice, just to look at the total mortality for the moment, the first set of data, that there's variation across countries. Most of the estimates are to the left, indicating improvement. In fact, the bottom line represents the overall data across all countries. But there's not always consistency. Iceland and Denmark, for example, have a result which is centered around 1, relative ratio of 1, indicating no benefit, as does the U.S., the bottom row. Is this real? Does this mean that there's no benefit of this drug in the U.S. or Denmark or Iceland? Why should these sites be different than Germany or Norway or Sweden? Where's the consistency? Well, let's look over to the next. Total mortality and hospitalization. Here, the U.S. looks like the world at large, as do some of the other countries. And the same for total mortality and heart failure hospitalization. So if we focus too hard on a particular subgroup, we may be getting results which are quite likely not true, not valid. There's variation across countries. In this case, unfortunately, the U.S. Food and Drug Administration really did focus hard on mortality rate in the United States, and actually, and disappointingly, never approved or labeled the use of this drug for reduction in mortality, despite the fact that the trial was terminated early for a 34% reduction in mortality. Was the FDA right or wrong? Well, who knows for sure, but there was not the consistency within this trial, and there was 2 other beta blocker trials in heart failure at the same time that did not see this anomaly in the U.S. mortality results. Most cardiologists would believe that the beta blockers work just as well in the United States as they do in the other countries. But by focusing too hard on a subgroup, we may have made a wrong decision or a wrong recommendation. Nevertheless, it's a challenge either way to focus really hard on a particular subgroup and come to conclusions. PRAISE 1 is a study that I would like to remind you of. This was amlodipine versus placebo, randomized, double-blind, placebo-controlled in a population of heart failure patients. It was stratified by etiology, ischemic versus non-ischemic. 1,153 patients were randomized. Overall, there was a strong trend for a benefit in the mortality curves for those patients on amlodipine. But the p-value was 0.07, not quite significant, not quite strong enough. However, if one did the interaction test comparing etiology versus outcome, you found that there was a significant interaction of etiology with outcome, p0.004. Further examination suggests that the ischemic group had a p-value that was not significant, but the non-ischemic group had a p-value of 0.001. Conventional statistical thinking would say, okay, you tested for interaction, you had a significant p-value that says there was definitely a difference in the outcome based on etiology, ischemic versus non-ischemic. Therefore, one should break the data down into two pieces, look at the ischemic group alone and the non-ischemic group alone, and one would be tempted to argue that there was a significant mortality benefit in the non-ischemic group. In fact, the p-value was 0.001. If you looked at the results by the ischemic subgroup, you can see that the mortality comparisons are nearly identical, but in the non-ischemic group, quite an impressive difference. The investigators debated what they should do about these results, but came to the conclusion that they needed a second trial. What was wrong with the previous thinking, or what was at least troubling, is that if they had any belief in how the results should have come out, they believed that the effect should be seen in the ischemic subgroup, not in the non-ischemic subgroup, because the results were opposite to what was expected. So to confirm this, quote, predefined subgroup, ischemic versus and non-ischemic, they repeated the trial in the non-ischemic group alone, became known as PRAISE 2. Again, same protocol, same kind of patients, just looking at only the non-ischemic group, and it was randomized, double-blind, and quite large, 1,653 patients, with mortality as the outcome again. Contrary to the previous study, the mortality outcome in the non-ischemic group had a relative ratio of 1.0, no effect. So one could have followed standard statistical reasoning from PRAISE 1 and said there is a significant mortality result in the non-ischemic subgroup. However, trying to repeat that result led to an outcome of a relative risk of 1.0. This illustrates the risks of conclusions of subgroups and the need to always replicate whenever possible. So the three views on subgroups that some people discuss is one should just ignore them and never do them, just only analyze overall. The second plan is to define these subgroups in advance and don't mine or dredge the data. Third is do the kind of subgroups you would be tempted to do, but always review them with caution and rarely make conclusions on the basis of any particular subgroup. Regardless of which approach you take, subgroups are challenging, and especially if you get curious about them after you've seen the outcome data. What I've tried to discuss with you is some issues that one needs to keep in mind in analyzing the result of your trial. As I indicated earlier, you could have the perfect design, the perfect conduct, but in the analysis stage, you could really introduce bias into the results and into the interpretation, something that we all want to avoid. So please keep these issues in mind. The intention to treat principle really is important to account for all patients and all events. Whenever you see papers or studies published that don't report all the events, you need to inquire as to what the missing data, missing patients might have added to the results. If it's your own study, please avoid the temptation to withdraw patients based on compliance or eligibility or any other criteria you might apply that took place after the trial began. Thank you very much.